Hi all, welcome back to our channel My Bio Notes. So in this video we'll be discussing about linkage and recombination. So before going into the video, please subscribe our channel and also click the bell button for more updates from our channel. So come, let's see about the concept of linkage and recombination. So what is linkage in genetics? Is that the closeness of genes to one another on the same chromosome. So if you take a chromosome, how the genes are close enough. So if you take this part, this is a gene, let's take this is an A gene, and let's see, it must be on the same chromosome, right? So let's say this is a B gene. So this two, A and B, are close to each other. And if you take this gene as a C gene, so when compared to the closeness of an A and a B gene, C is not that close. So A and B are close to each other, but C is further apart. So this A and B will be linked. So that they are that they say as the closeness. So they will be associated with each other, the A and the B gene. So that you call it as a linkage. So in linkage, this linkage, we say it is located on the same chromosome, and also here the gene, it will be expressing some character, right? So this A gene will be responsible for one character, say character 1, and B gene will be responsible for a character, it can be character 2. So this A and B character will also be inherited together. So that's why we call it as a linkage between these two genes. So example, you can say that eye color and hair color, they tend to be inherited together, always they are inherited together. So we can take that as an example. So since these genes, so phenotypically, if you see, say C1 is a character for eye color, so eye color and hair color, so these both characters, it will be the same in the parent and the offspring, because these genes are close to each other and they have a linkage in the chromosome. So this will be same in an offspring and a parent, eye color and hair color, because of these genes, because of the gene linkage. Just like see how the chains are linked. So in the same way the genes are linked in this chromosome. So this is about linkage. So even a blonde hair, you can say a blonde hair or and a blue eyes. So if it will be the same in the parent and the offspring, we can say it is due to the linked genes, because the genes are associated with each other. Or you can say brown hair and brown eyes, if it is the same in the parent and the offspring, you can say it is due to the linked genes present in the chromosome. So this is about linkage. So next we'll discuss about recombination. So what is recombination or crossing over? So recombination, so it is a process or act of exchange of genes. So here what is exchange? It is the genes which are exchanged between the chromosomes. So yeah, we are talking about the chromosomes only in genetics, and in this case also chromosomes plays a major role in terms of genetics, right? Because it contains the main genetic elements, the genes for the inheritance of the traits or the characters. So here the recombination is the process of exchange of genes between the chromosomes. Okay, so here you can see the sister chromatids and the exchange by recombination. This process you call as recombination or crossing over. So you might have studied in cell cycle, right? So this crossing over or recombination occurs in the meiosis stage of the cell cycle during the Pachytene stage. So during that, this crossing over or recombination occurs. So why we are talking about this here is that, so this recombination or a crossing over, it is a genetic variation. So the offspring will be varied from the parents due to this recombination or crossing over. So due to this genetic diversity, there will be added advantage for the offspring. The character which is not found in both the mother and the father, you can see a new type of character in the offspring. So how that adds an added advantage to the offspring. So the new trait, it can also be an advantage, like it can help to survive the offspring in a stressful condition. Both the parents may not be able to survive that condition, but the offspring will have this new trait and it will be able to survive any stressful condition. So that's the importance of recombination. So here you must know this concept of recombination before going into the Morgan's experiment, because these two terms, linkage and recombination, are the two terms that he will be discussing in his experiment. So again the different genetic combination and ultimately to the formation, this different genetic combination and formation of unique gametes with chromosomes that are different from those in the parents. So yeah, that's what I said. So this genetic recombination, it creates the offspring, the gametes, they say here as gametes. So from the gametes the offspring will be formed after fertilization. So this unique gametes, it is due to this recombination or crossing over. Without this event, you can't get a new trait or a new offspring with a different character. So this is a non-parental gene combination. So this trait or a character which is inherited from this recombined gene, you can't find it in parents. So it is a non-parental gene combination. So hope you are clear with the two terms, that is a linkage and recombination. So now let's move on to the Morgan's experiment. So this Thomas Morgan Hunt, what he did is that he did dihybrid crosses in Drosophila. So dihybrid cross we have already discussed in right, using two traits. Dihybrid, di means two. So already Mendel did this dihybrid cross, we have already studied, but here Morgan did this cross in a different organism. The organism name is Drosophila. So why he did this experiment is that to study the sex-linked genes. So what we call here as sex-linked genes, so some genes which are responsible for the characters or a trait, so that will be present in the sex chromosomes. So what you call it as a sex chromosome? So sex chromosomes are XX in case of a female and XY in case of male, right? So in this chromosome some traits or characters will be expressed from this chromosome. So the X chromosome will be long and the Y, I don't have a picture of that, and the Y chromosome, it will be short. Okay, so some genes here in this is X chromosome, this is Y chromosome. So if it is XX, it's going to be a boy, and XX is girl and XY is boy, right? So here it will be having some genes in this chromosome. Yeah, chromosome means it will be having some genes, right? So you can name some the genes by any alphabets. So in this genes some characters will be expressed. In the same way in the sex chromosomes also, that is the X and Y chromosomes also, the characters will be expressed and some genes will be present. So that he studied in this experiment. So he studied the sex linked genes. So the genes present in this sex chromosomes he studied, and it is similar to the crosses by Mendel in peas. Already we have learned this. You can refer my previous videos. So the same thing, the same experiment he did. So this Morgan and group, they were aware that the genes were located in the X chromosome. So now what we are going to see in the experiment, the gene which is responsible for the trait is present in the X chromosome. Here the eye color it is present in the sex chromosome. So two genes of dihybrid cross were situated on same chromosome. So if you take if two genes, if they are present in a same chromosome, it results in proportion of parental gene combination were higher. So the result is that the parental gene combinations will be higher. Why we are saying this is that since they are situated in the same chromosome, the two genes of a dihybrid cross, more of the parental gene combination will be seen in the offspring. So that we'll be discussing in the detail. If you can see it in a picture format. So low recombination, when we will see low recombination, so recombination, now the concept will be clear for you, right? Recombination or crossing over, it is the exchange of the genetic material. Okay, so when there is low recombination is that when the genes were tightly linked in chromosome. So imagine that, so when a crossing over has to occur, we have two homologous chromosome. During meiosis stage the crossing over will occur, right? Patch 18 stage. So we'll be having two chromosomes, right? So here what happens is that during the exchange of the genetic material, which we call by the term as recombination, when the genes are tightly linked, like in the chain, when their chain is tightly linked with each other, so it becomes difficult for that genes to get split, right? So when there is low recombination is that when the genes were tightly linked in chromosomes. So example, they give us white and yellow genes here in this experiment that we'll be discussing later. So how much low recombination is that? They have given it in percentage. It is 1.3% only recombination is there. When why such a low percentage of recombination is that? The genes are tightly linked. During crossing over also it does not get split, it doesn't get separated, the genes. So that's why there is this much low percentage of recombination. So when there is higher recombination, when the genes were loosely linked. So when the chain is loose, it's easy to separate the genes, right? So it resulted in higher recombination when the genes are. So there will be exchange of genetic material between the two chromosomes. So the genetic material will be exchanged, then only we will be getting new traits, right? So when the genes are loosely packed, it becomes easy. But when the genes are tightly packed, it becomes difficult for the recombination to occur. So you can say the example here as white and miniature wing. So what are the traits? White and miniature wing. Here wing trait. Here white and yellow genes. Okay. So here no, the recombination is higher. You can see 37.2% of recombination. It's very high. When compared to 1.3%, which is very low, this is very high. So that's what Morgan said. So you must know the terms linkage and recombination. So it is due to the linkage. Linkage only determines the recombination percentage. So how the recombination is going to be. So how the genes are associated in a chromosome is going to determine the recombination or crossing over event. So thereby how the traits are getting inherited is also determined. So you can see in the picture, right, the same picture in your book. So here you can see the cross A, here you can see the cross B, the male and the female are given here. So here they have given us chromosomes. Why as chromosomes? Till now Mendel experiments and all, we saw it as capital means dominant, then small letter means we saw round means capital R you had to say, and all round yellow we saw. No, why now in picture format? Why they have given in chromosome is that now the microscopy has developed during his period. Now you can see the genes in the chromosome itself. So it's easy to see the genes in the chromosome here. So they have given it in the form of chromosomes. So here you can see that now we saw some characters they resulted in low recombination. So that yellow and white, this is wild type, that is the red-eyed one, red-eyed fruit fly chromosomes they have shown here. They have given it the genes as Y plus and W plus, small y plus and W plus, and here small y W. Yellow and white they have given, that's the parental female and the male, he crossed and he got the offsprings in the F1 generation. See, there was a wild type which is the red-eyed fruit fly, and yellow and white-eyed was present. So next again, if you see in the F2 generation, because the same cross, so just imagine the dihybrid cross done by Mendel, but they have given it in the chromosome format. That's the difference. So the parental type here, because here we will get plenty of offsprings, because a fruit fly is able to lay hundreds of eggs. So here the parental type they have given as percentage was 98.7%, and the recombinant type was very low. It was 1.3% only. So you can see the gene chromosome and the genes here, gene combinations. And coming to the cross B, so the male and the female genes here, the wing trait was taken, right, white and the miniature wing, and the wild type is given, so it will be a big wing. So how the parental type and the recombinant type was expressed here? So here the recombination was very high, it was 37.2%, but the parental it is 62.8%. But compared to this cross, here we got a high number of recombinants. Okay, because see how the genes are arranged. Here it is very close in these two chromosomes, but here these two genes are far from each other. So during the recombination is easy for these genes, the recombination or crossing over, you can see. So that's why we got a high number of recombinants. So next, Alfred Sturtevant. So Alfred is a student of Morgan. So he said that the frequency of recombination between gene pairs on same chromosome, by this data, if you have the frequency of recombination between gene pairs, we can determine or we can measure the distance between genes. So that he found out, Alfred, who was Morgan students. So it is a good idea, right? The frequency, how frequent the recombination occurs between a gene pair. If you know the data, you can measure the distance between the two genes. So how distant, if you take a chromosome, how distant the two genes are. So the distance can be measured. So this distance, in turn, we can map this distance information gives the position of the chromosome, position of the genes in the chromosome, where the genes are exactly located. So the position of the genes can be mapped by this data. So this led to the development of genetic maps. So what are genetic maps? It's like the normal map that you see. So this genetic map is the starting point in sequencing of whole genomes. So whole genome sequencing, no, this genetic map is the starting point, and the example you can say as the human genome sequencing project. So that you might have studied, and this genetic map is nothing but the normally where the gene is exactly located, that you can see in a genetic map. So the genes are mapped, its location. So that's it. Thank you all for watching my video. If you like the video, do subscribe the channel and also click the bell button for more updates from our channel. Happy learning to you all. Thank you.